இது நம்ம ஐயா கோச்சிங் சென்டர். வாங்க படிக்கலாம்.
இந்த வீடியோ உங்களுக்கு பிடிச்சிருந்தா லைக் பண்ணுங்க, ஷேர் பண்ணுங்க, சப்ஸ்கிரைப் பண்ணுங்க. Thank you.
Today let us see dihybrid cross. What is meant by dihybrid cross? That is a cross in which the study of inheritance of two pairs of contrasting characters. So in this cross, two pairs of contrasting characters are taken by Mendel. Contrasting characters. What are the contrasting characters Mendel has taken? Color of cotyledon. Color of cotyledon, that is yellow. Then green, yellow and green small y. And then seed form. Another character is seed form, that is round and wrinkled. The next one is yellow and round characters are dominant. There is yellow and round dominant. Then green and wrinkled recessive. When Mendel crossed this yellow and round with the green and wrinkled plant, all the F1 progeny are round yellow. When the F1 plants are self-pollinated, then 4 kinds of plants are found in F2 generation. What are the 4 kinds? That is yellow round, then yellow wrinkled, then green round, then green wrinkled. Green wrinkled. So these are the 4 types of plants which when F1 progeny is self, 4 types of plants are got, and the ratio is 9:3:3:1. This is dihybrid ratio. Now we will see it in the checkerboard. Okay, let us see parent, that is round yellow, capital R, capital Y. Then wrinkle green, small y. So this parent is round yellow and wrinkled green. See the gamete, it is formed, and here the gamete is small y. See? So the F1 progeny is capital R, small r, capital Y, small y. This is what? Round yellow. When this F1 progeny is selfed, capital R, capital Y, small y, capital R, small r. When it is selfed, first, then now we'll see the offsprings through checkerboard. See, this is a male and this is female. Now the gamete is capital R, capital Y, capital R, small y, capital Y, then small r, small y. Same here, capital R, capital Y, capital R, small y, capital Y, and then small r, small y. Now you see the checkerboard. This is male gamete and this is female gamete. And the offspring which got us given in this square. So we'll see now. See, this is round yellow and this is round yellow, because which is dominant? Round is dominant and yellow is dominant. So round yellow, and this is also round yellow, and this is also round yellow. This, and then this one and this one. These are round yellow. See, just you can draw like this. See, this one here, like this. So how many round yellow? Totally 9 round yellow. How many? 9 round yellow. 9 round yellow. The next one is round green. You can see here round green. See, this one is round green. You can use a black color. This one is round green, and then this one is round green, and this one is round green. See, like this. These 3 round green. So 3 round green. The next one, see, this one is wrinkled yellow. This one, this is wrinkled yellow. How many? 3 that also. 3 wrinkled yellow. Wrinkled yellow, and only 1. This is what? Only 1 wrinkled green. Wrinkled green. So the phenotypic ratio is 9:3:3:1. And what is the genotypic ratio? Genotypic ratio is, that is genotypic ratio. This is what ratio? Phenotypic ratio. Phenotypic ratio. And the genotypic ratio is 1:2:4:1:2:1:2:1. This is the genotypic ratio. So what is the phenotypic ratio? 9:3:3:1. Genotypic ratio is 1:2:2:4:1:2:1:2:1. See how many? 1, 2, 3, 4, 5, 6, 7, 8, 9. Previous class I have explained about the formulas. That is, to find out the genotypic character, the formula is 3 to the power n. What is the n? Heterozygous. How many heterozygous pair here? See, you can see here only 2. 1 and 2 heterozygous. So 3 into 3 to the power 2, that is equal to 3 into 3 is equal to 9. So 9 genotype. You can, so in F2 generation, you got how many? 9 genotype. Okay. See, round yellow and wrinkled green are parental. And these are new combination. These are formed by means of new combination. Produces the result of new combination, that is round green and wrinkled yellow. Round green and wrinkled yellow. Mendel's dihybrid crosses inheritance of 2 genes. After this experiment, he framed another law. He got conclusion, that is law of independent assortment. Law of independent assortment. Now we'll see this law of independent assortment. This law of independent assortment, it states that when 2 pairs of independent alleles are brought together in F1 hybrid, that is F1 hybrid, we got what? This one. The F1 progeny is round yellow. Round yellow. So when 2 pairs of alleles are brought together in F1 hybrid, then they show independent dominant effects. And the law of independent assortment is also applicable to only those factors or genes which are present in different chromosomes. At the same time, during gamete formation, that is during gamete formation, the 2 factors of each trait, they assort random and independent of the factors of the other trait at the time of meiosis, that is gametogenesis, and get randomly as well as independently rearranged in the offspring. Okay, then the law of independent assortment is applicable to only those factors or genes which are present on different chromosomes. For example, different chromosome means that is like this, capital Y, small y. So, Mendel's law of independent assortment is applicable to only those factors or genes which are present in different chromosomes. Then these gene pairs did not show. Exception is linkage. Exception is what? Linkage. Same like that in law of dominance also. The exception is that is incomplete dominance and also codominance. The law of dominance. Law of dominance is also exception. It shows exception in, that is incomplete dominance, and also codominance. Law of independent assortment has also exceptions to which one? Linkage.